Now, come on, let us give a big hand to Prof. Jan Ganon. Goedemorgen.
Thank you. Thank you. And wow, congratulations on the pronunciation of the very difficult Dutch. Very well done. Good morning, everyone. It's a great pleasure to be here. So thanks for the invite and thanks for the great event we're having so far. And I'm here indeed to talk about, I'm a microbiologist, so I will just continue what previous speakers have talked about, the gut microbiome, and the gut microbiome as an important health factor for our overall health, specifically focusing on C-section. So as a microbiologist, of course, I have to show this slide to get you all again in the mind of the gut microbiome. Complex system. I think we sometimes underestimate how complex it is because there's hundreds of species that all interact between each other, but also with us as a host. They're impacted by the immune system, they're impacted by what we eat. So a lot is happening in the gut. And this is of course very important in early life where the whole system has to establish and develop itself to be the digestive tract that we need for the rest of our life. And if you look at indeed evolution, we see that human milk has evolved to indeed not just only feed the baby to grow, but human milk also has evolved as a kind of an optimal gut microbiome modulator. It's controlling the colonization of the gut, it's repressing pathogens, so it's also, I think, jumping in for the immune system that is yet immature, the immune system of the infant. And human milk really protects the infant from pathogens while the immune system is still developing, because there's lots of things in human milk, like the antibodies, antimicrobial peptides, that inhibit pathogens. But also in human milk, you find substrates, complex sugar molecules that the infant cannot digest. The infant cannot use those sugars to grow, but those sugars are just in human milk to actually support the growth of beneficial bacteria in early life. And why in evolution would we put so much energy in these sugars to put them in human milk if they would be of no use? So I think it really shows the importance that in evolution humans have tried and established this support of the symbiosis in different ways. So there's immune cells in human milk, there is these oligosaccharides, very different structures, complex structures, showing that we probably need to support different types of bacteria in early life. And if you look around the globe, the microbiome of infants is quite similar. So whether you have a baby from South America, Asia, Europe, this general concept of human milk oligosaccharides and the quick colonization by bifidobacteria and Bacteroides is really a global concept. Well, we now also start to understand that even also lipids, lipid structures, they can actually also impact the microbiome. So in human milk, don't just think of it as a growth engine for the baby, but also as a very important engine to drive the gut microbiome development. And you also see that when you look into the composition, that I think this is a very clear example of a symbiosis. Of course, the bacteria benefit from us because we protect them, that we give them a nice environment in the gut where they can grow. We protect them against other species, but we feed them even. We give the human milk oligosaccharides to feed the bifidobacteria. But the bifidobacteria then also in return give us back certain aspects of colonization resistance, production of vitamins, so it's really a win-win. And when you look at the composition of human milk, you see these very complex human milk oligosaccharides structures. And then if you look at the genetics of bifidobacteria, for example, you see that these bifidobacteria have really evolved to optimally utilize the human milk oligosaccharides. They have all the genes that explain why they can use human milk oligosaccharides. They have transport systems to take up the human milk oligosaccharides. They have all the enzymes to degrade the human milk oligosaccharides. So it really shows that bifidobacteria are specialists on human milk oligosaccharides. And I think in evolution this really has co-evolved, because we see that human milk is very high in these human milk oligosaccharides. It's even more human milk oligosaccharides in human milk than there is protein in human milk. We would think maybe that protein is really a growth driver and the most important ingredient in human milk. But there's even more of these useless human milk oligosaccharides that the baby does not use, but it's really to feed the microbes. And we don't see this in other milks. For example, cow's milk, often the basis, of course, for nutrition in early life, does not contain human milk oligosaccharides. So it seems to be quite specific for certain species of mammals, and humans, they really have very high levels of these prebiotic fibers. Well, how do the bifidobacteria then use these, and why is it important? Well, we think it's not just to feed the bifidobacteria, but bifidobacteria, as mentioned also before, are a critical species. They are a keystone species, we think, because they shape the early development of the gut. They create an environment in the gut lumen that is important, because the bifidobacteria, they can ferment the human milk oligosaccharides, which makes the environment more acidic, which inhibits growth of many pathogens. It makes the gut lumen more anaerobic, allowing the growth of strict anaerobic bacteria, which are all essential processes to get the right gut microbiome going. And when the bifidobacteria degrade these human milk oligosaccharides, they spill also leftovers of the sugar moieties, they produce short-chain fatty acids, and all these things they produce actually can be used by other bacteria. So also bifidobacteria cross-feed other bacteria to really start and shape the development of this complex microbial ecosystem. Well, I think this is quite an interesting study from Israel, which also shows that during pregnancy, the body apparently seems to prepare for delivery also from a microbial perspective. So at the end of pregnancy, we see that the levels of bifidobacteria seem to increase in the gut microbiome of the mother. So it really seems that the biological system is getting ready for delivery, improving also the transfer of beneficial microbes from mother to offspring. Because these bifidobacteria need to transfer bacteroid, strict anaerobes. They're not in the environment, so they really have to come from other humans. And of course, the most obvious one is the mother, because of course the transfer that can happen. But then, when an infant is born sterile, it's still a sensitive process to get this process going, to make the bacteria grow in the gut. And there's actually quite a few things in modern society that can really impact this normal gut colonization. And they've been mentioned already before this morning. Of course, we need to make sure that the mother has a healthy microbiome. I think it's maybe a little bit understudied or has not enough attention, but I think it's really important to make sure that when women are at childbearing age, that they have a good microbiome. So if we can support that through a healthy diet or healthy lifestyle or through biotics, I think it's something to consider, because the mother is a very important source of microorganisms for the offspring. But then also indeed in modern society, antibiotics are used quite a bit, of course. They have saved many, many lives. But at this sensitive stage where the system is developing, antibiotics also can have a significant impact on this developing gut microbiome. Preterm labor has been mentioned, and of course also C-section, and that's really the topic that I want to focus on. And sometimes, we can even consider that an elective C-section is kind of a triple hit, because these infants are born pretermly by definition. They're born too early. They receive antibiotics in almost all cases, and they're born by C-section. So there's really three big hits that definitely have a huge impact on the developing gut microbiome. And this is important, and okay, what is the real window is, I think, always a discussion, because you have the developing system, you have the developing gut, the developing immune system. What is the biggest window to impact the immune system? But anyway, we also want to always improve the gut microbiome, because it's not just important in the first weeks of life. Gut microbiome, we see more and more, is important, and gut dysbiosis is very often linked to all kinds of diseases. So we really have to find the right tools to lift up the microbiome and make sure that it is a stable and healthy gut microbiome. Well, today I will really focus on data we have now with a symbiotic. A symbiotic, as already mentioned and explained before, is a combination of pre and probiotics. In this case, a very specific one, a mixture of GOS and FOS, as also mentioned already, studied more than now for 2 decades, as being a very good prebiotic mixture that mimics very much the function of human milk oligosaccharides, and a very specific probiotic strain, Bifidobacterium breve M16V, which we think is really very suitable for young infants. First, a bit on the prebiotics. I already mentioned this. Human milk contains a very high amount of prebiotics, even more of these substrates for microbes than there is protein. Again, this is lacking in cow's milk, which of course often is the base for infant formulas. So for this, 25 or 30 years ago, this mixture of prebiotics was developed based on what we see also in human milk. The structures in human milk are complex. They have very different sizes. You have small ones, you have big ones. Thinking that the small ones are of course first fermented, the more larger and complex ones are fermented more down the tube. So it really provides a substrate for bacteria all over the GI tract. And this is mimicked also in the GOS-FOS mixture, where we have galacto and fructooligosaccharides that stimulate different types of beneficial bacteria, but also are easily fermentable and a little bit more hard to ferment to really feed the whole GI tract with this fermentable fiber. And the levels are very similar, the prebiotics used, as we find levels also in human milk. This also was shown, this is quite a robust, scientifically supported prebiotic mixture with lots of evidence, clinical evidence, and showing the benefits for gut health, stools, immune system, and of course the microbiome composition. So many studies, and these are now also recommended, that if indeed we cannot breastfeed, that indeed this is a very good substitute for that. Then the probiotic, I think it's maybe a little bit new, and you can imagine that if you have an infant born from a healthy mother and the infant is inoculated with the right microbes, breastfeeding or nutrition with prebiotics will further shape the microbiome. But in the cases that we mentioned just before, where there's really a big hit on the microbiome, whether it's antibiotics or C-section, just feeding the microbes with substrate is not enough. If the right microbes are not in the gut, if the microbes are not in the system, you can feed them with prebiotics or fiber, but it won't work because they're just not there. So in those cases where we really are lacking the initial inoculation of beneficial microbes, we probably need to put them back into, and probiotics of course is a very good tool to do that, using the live bacteria in nutrition to support the gut microbiome. And this is also inspired by human milk. I think there was in the past a discussion whether human milk is sterile. People assumed maybe that human milk is sterile, but now there's also consensus that human milk is not sterile. It always contains low levels of bacteria, also sensitive for antibiotic use. So also this is another case. If these mothers use antibiotics, you will also impact, of course, the microbes in the milk. And we see specific species, and this was also the inspiration for us to have a focus on certain types of bacteria that we could be using as probiotics. And also explained already from the Italian lady from Bari that it's very essential to be very specific on what we talk about, because not all bifidobacteria are the same. If you look at the ecology of bifidobacteria, we find that certain bifidobacteria we actually only find in certain animals. Certain bacteria we find even in the environment, in insects. But also, if you look at humans, even the bifidobacteria that we find dominantly in adults is different from what we find in young infants. And this is probably has to do with the environment of the gut, the diet we have, but also the optimal co-evolution of these types of species with the host. And if you then look at what we find in breastfed, healthy human infants, we find this very specific infant type of bifidobacteria, like Bifidobacterium breve, Bifidum, Longum infantis. This is really found around the globe, the most dominant bifido species in breastfed infants. What we see, of course, is also use of probiotics of these other species, but we think they're not always really human resident bifidobacteria. And it's important to stick to the symbiosis that we see in nature, to also use then the most natural species of bifidobacteria, which was a challenge because these bacteria are very sensitive to oxygen, and it's very difficult to get them into a nutritional product. So it took quite a bit of technological breakthroughs to be able to put this strict anaerobic bacteria into a nutrition like an infant formula or a supplement. But this is now feasible, doable, and we can also use these types of bacteria, which I think was very good news. And then we can also, of course, study the effects of these types of bacteria, and that we have done. We think that this is a concept that is very close to what we can learn from human milk. We have a lot of safety data because, of course, this is a first question. In sensitive infants, in sick infants, can we use live bacteria safely in our clinic? Well, I think there's a lot of evidence, even in preterm infants, that we can use these types of bacteria safely. And we screened amongst many other different types of probiotics in preclinical research. Years of work, just now one box in a slide, but it's a lot of work where we compared and then where we showed that actually this Bifidobacterium breve M16V was, we think, the best probiotic to take because it's superior in many of our preclinical assays, both in the lab but also in animal research. And all this data now indeed also has supported the registration in the US, in China. It's well approved by regulatory bodies around the globe. And of course, what triggered us to really also use this combination is synergistic effects that we saw in our preclinical research. So in some of our animal models, for example, this is just an example, we see that indeed if you use the combination of the live bacterium plus its substrate, so you give the live bacteria and you give it a lunchbox, it's more efficient in many of the models than just using one of the concepts. And I think it is because the prebiotic is indeed a growth substrate for the probiotic, but also you have a mixed effect because the prebiotic will impact the gut microbiome composition and activity, its substrate for producing short-chain fatty acids, and that will also make the probiotic more effective. So this is really a concept that we then tried in different target groups, but today I will really focus on the data we now have in C-section born infants, because that's really also the focus of today's session. And C-section, well, it was already discussed before, it's really not an underestimated number here. I think it's average numbers, but I know that in some parts of the world, in private clinics, in certain parts of the world, it's more or less the standard to be born by C-section. So a great need, and this is then also what we studied. Can we support the more normal gut microbiome development with these kind of symbiotic concepts? Well, this is a Julius study. You already had a sneak preview of the data of the study. It's the same study that you just saw before. It's a study in Southeast Asia where we indeed compare vaginal delivered infants to 3 intervention groups, just a placebo group, a control group, a prebiotic group, and then the symbiotic group. And we were quite lucky. We were able to take longitudinal samples, quite a few, of which we could then also do all the omics analysis that today we have available. We did metagenomics, but also metabolomics to just not understand who is developing as a microbial community, but also what can this microbial community produce, which is relevant for human health. And indeed, we were very happy to see that indeed we can correct at least the levels of beneficial bacteria very early on. And it's, I think, quite surprising results that already after 3 to 5 days, which often is the first fecal sample produced, that you already see high levels of beneficial bacteria when an infant is born vaginally or when it's receiving the symbiotics. And it also shows that our concept that you cannot rescue the beneficial bacteria with prebiotics alone is also confirmed. The 2 lower lines, of course, the control group and the prebiotic group, you cannot rescue the beneficial bacteria so efficiently as you can with symbiotics. Well, then of course we often got the question that, okay, if you give symbiotics, these infants, the only thing they will have in their guts is the M16V, so you get a kind of a monocolonization of infants with the probiotic. Well, this is really not what happens, and this is what you see in this plot, where we looked with metagenomic sequencing at the Shannon index, so it's an indicator for gut microbiome diversity. It's quite a big topic in science these days because people really believe that gut microbiome diversity is very important for a healthy microbiome. And we see actually that also the symbiotics drive also this diversity. And I think this relates to what I said in the beginning, that bifidobacteria can stimulate the development of a microbial ecosystem by cross-feeding other species, by creating an environment where these types of bacteria can really thrive very well. And we dived in a lot more detail, and I'm a microbiologist, so I will not bother you with all the microbial names, but what you see in general is that when an infant is born naturally, the system is going in a certain direction quite fast. So it goes to anaerobic, lower pH, driven by, of course, bifidobacteria that utilize the human milk oligosaccharides. And this allows also the colonization of strict anaerobes. And these strict anaerobes are really important because strict anaerobes are the ones that cannot tolerate oxygen. They need a certain environment to grow. But then they start to produce also butyrate. So butyrate is really produced by strict anaerobes. And I think we all know that butyrate is very important for gut integrity, for gut health. So it's important to get this system going in this direction. Whereas in C-section born infants, we see much more of the facultative anaerobes, and we see less of the strict anaerobes. But it seems that the system is becoming less anaerobic, and it allows more the growth of bacteria that can grow in the presence of a little bit of oxygen, which are quite often enterobacteria, say Klebsiella, bacteria that produce LPS, endotoxins, that can cause this gut inflammation. And we actually think that by causing a gut inflammation, you will still have more oxygen in the gut, allowing even more the bacteria that can deal with oxygen to take the scene. So it's like a switch. You make it very anaerobic with strict anaerobes, bifidobacteria and Bacteroides, or you stick to the more undesired dysbiotic microbiome. But with symbiotics, we can actually really push the system into what we also see in a vaginal livid infant. It becomes anaerobic, it becomes lower in pH, and we see very similar microbial patterns with the symbiotics. And well, I will not go into the details. This is published in the paper. I think this one's in the supplement, so if you really want to have a look what we think mechanistically happens, I suggest that you look at this paper where we try and explain these graphs. But this really explains how a system can shift from, let's say, fermentative microbiome with a lower pH to, let's say, an undesired dysbiosis, and how symbiotics can drive the system, in this case, really to the left, which is very comparable to a healthy breastfed infant gut development. And because this microbiome development, of course, is associated with an aberrant also immune development, we were very happy to see also in this study that actually already, whether this is a relatively small study, we see reduction of immunity-related disorders. And this is really the skin symptoms that we see quite often in C-section born infants. That the symbiotics are really able to reduce the proportion of infants with skin disorders, and it becomes very similar to what we see in the natural vaginal delivered infants. Well, I think one study is no study, is what some people would say. So we're now happy to share that we also have data from other studies. This is a study from China, quite a big study where we actually looked at tolerance of a new formula, but also comparing symbiotics with prebiotics. And of course, if you do a study big enough, you will always have also enough numbers of infants that are born by C-section, and that's also the case in this study. And also here we actually see that in C-section born infants, we can increase the level of beneficial bacteria with the symbiotics. On the right again, we see the diversity of bifidobacteria, also confirming the first data from the first study. And in this study, we see that indeed, and the bottom two lines, the dotted lines of the C-section born infants, that indeed we can also, in this case, we can increase the level of beneficial bacteria in C-section born infants in the Chinese cohort. Well, here we did not only look at bifidobacteria. I think it was also mentioned before that another typical bacterium that suffers from C-section delivery is Bacteroides, and this is because Bacteroides is indeed one of these very strict anaerobes. It needs certain conditions to be able to transfer to the offspring and to grow. And we see actually that also with the symbiotics we can increase these Bacteroides, and I think this also shows that by creating a right environment, you allow a better colonization of other species. And this colonization of Bacteroides is not just during the intervention, but we also see that later on, after a year, these Bacteroides species are at higher levels, than without the symbiotics. So the bifidobacteria impact other species as well, and these effects are not just during intervention, but also after intervention. And then the latest one, which I think was also an exciting study from Hong Kong, it's still running, it's a real-world evidence study. Always a bit exciting, of course, to see what happens in real life, because you do not really control the study, as we scientists would like to do. But we just get feedback, we take samples from real-world use of the symbiotics. And also in this study, we were able to confirm that in this setting, with symbiotics, we can really rescue the colonization of beneficial bacteria in C-section born infants. Well, then to some important messages. I think we all agree it's quite clear that human milk is the best nutrition for babies, but not just for babies. Also, human milk is really the best nutrition for a developing microbiome because of all the bioactive components that are in there. Well, it's very clear, and I think I saw in the previous talk, there's 100% consensus that C-section indeed impacts the initial colonization, and that these infants have delayed establishment of this anaerobic, slightly acidic gut environment, which is linked to gut inflammation. Human milk contains things that we can learn from, things like biotics, prebiotics, probiotics, and that early supplementation with symbiotics, and I think, again, we need to be very specific. The data now is there for this very specific mix of GOS and FOS and Bifidobacterium brave and M16V can really restore the colonization with Bifidobacteria. And that these symbiotics can really improve also biomarkers that indicate a more healthy gut and immune development. So thank you very much for your attention, and please remind that we do not only feed the human, but that also in infants we really need to take care of taking the right nutrition to grow the right microbes. Thank you.
Well, thank you so very much, Prof. Jan Knol. All right, ladies and gentlemen, once again, I would like to say a big thank you to Prof. Jan Knol. Please give Prof. a big round of applause. Dank u wel. All right. So at least it's like from Prof's session, I get to learn